Hi, everyone. So as Jesse said, I'm Matt Mulhern from AbbVie Pharmaceuticals. I'm a principal research scientist. So let me give you some of my experiences, with working with these data-rich experiments and how this is leading to some data paralysis inside of pharmaceutical development. So the overall trend inside of pharmaceutical development is that we're running these data-rich experiments. We're bringing together a lot of different information from a lot of different sources, both online and offline. And what we're trying to do is make these correlations between the conditions used in these experiments, the parameters in these cases, and the results, whether that result is impurity data or maybe NMR data. But as we're generating these large data sets, we start to run into this problem. What did we actually learn? And how do we even interpret these large data sets? This data-rich experimentation can very easily become data paralyzing. We gather all this information and then we try to think of a use for it later on. How did we actually get to this state? Well, just in a blink of an eye, we've gone from having mercury thermometers that you would stick inside of a reactor, and if you wanted to have data from that, you'd have to walk up to your fume hood, take a look inside, record that temperature into a laboratory notebook. Maybe it was even a paper notebook at the time. We've moved on to simple sensors. Maybe those were still being recorded then with a paper notebook through visual observation, or maybe it was tied into an electronic notebook in its first generation, which wasn't much more than glass over paper. But nowadays, we've moved on to things like, pictured here on the screen, these Mettler-Toledo reactors. They're gathering data from multiple different sensors and probes, and they're doing so every 2 seconds. So we start generating these huge data sets, but how can we still get it to something useful? So we need to capture that data from those reactors and still marry that to this offline data analysis, such as HPLC data. Current practice, we move to something like Excel or a spreadsheet, mostly because it's readily available. So this is an example here of just an Excel spreadsheet with all these different data points. So how do we get this to something that I can actually start diving into some details, but also get to something where it's manageable, something for visualization, because that's how we end up looking for our first round of information about the actual reaction itself. Counterintuitively, what we actually start doing in most cases is reducing the amount of data that we're working with. Maybe we were capturing data every 2 seconds throughout the reaction, but maybe we didn't need to be capturing at that data rate. So the first thing we're going to do is, as an example here, capture data that was only there every 1 minute. Perhaps we would even truncate this further to say, rather than looking at an entire reaction sequence, we're only looking at one portion of that overall reaction. The overall goal with this data reduction is to make the data set more manageable so that the processing on the computer in order to create our graphs and our visualizations doesn't take as long, and it's a little bit more interactive for us. But even after we've done this data reduction, we still have another part to our workflow, and that's bringing in our offline HPLC analysis. Could be any type of analysis really, but any offline result that we want to bring into these overall conditions that were captured from the Mettler-Toledo software. So in this case, I'm giving an example of some HPLC data. So we had the information about when the sample was taken, and then the impurity information that was generated. So now we finally made that correlation between our conditions and our results. So the last piece is doing the visualization. And this is where we can get into some of that analysis. We're looking for excursions in terms of some of the conditions that we're running. Maybe we had a temperature spike in the reaction. We can also start to look at those parameters and their impact on whether it's a starting material that's being consumed, products that's being formed, or maybe impurities as well. Were those impurities generated throughout the course of the reaction, or did they only start to form after the main reaction was complete? So that time course information is highly important, obviously, inside of the pharmaceutical industry. The other piece that we need to really focus on is the ability to transfer this information across scientists. Right now, most of our Chem ELNs don't do a very good job of letting us compare results from one scientist to the next. And that's where the beauty of Luminata really comes into play. It's that data accessibility, that ability to look at that single source of truth. And on top of that, we also have the ability to make use of the Spectris platform, and that involves Spectris Processor. Spectris Processor is able to read over 100 different data file formats. For us, that has a lot of importance because it doesn't matter then whether I use Agilent ChemStation software for my HPLC analysis or one of my colleagues uses Waters Empower system. In both cases, it's an LC system producing the same type of results. In the past, we couldn't bring those two results together except if we used something like a PDF or extracted data into some sort of a CSV and worked through a very complicated methodology. In both cases, with a Spectris platform, you convert these to a Spectris file, and then you can immediately go into Luminata with that type of data. We also can enforce a certain consistency of peak naming. If I call something dimer 1 and my colleague calls it dimer A, we suddenly have a disconnect. And inside of the Spectris platform, that disconnect can be resolved. So that is one of the other major benefits of having both Luminata and the Spectris platform together. So all those pieces come together in terms of the iControl tool itself. And for that, that's going to be the collaboration between Mettler-Toledo and ACDLabs that Joe DiMartino will be talking about next.